Okay, we should be live. Okay, we have a counter. Okay. Hello, everyone. Welcome to the session of Big Data Management. Before we start, we're just going to say Oracle first person this session. So Oracle has been an active member of the R Consortium since its inception, and they helped to establish the advantages of R, and we invite you to check their website for more information on that. So our first speaker is Roland Do. So the floor is yours. You can go ahead and start the presentation.
Thanks for having me here and excited to be here. I pre-recorded my talk and I will begin that shortly. This talk is about data version control for the targets R package. A lot of work that happens in R is computationally demanding. Everything from machine learning to Bayesian statistics to ETL. It's common practice to manage the complexity of a project in a sequence of notebooks. These notebooks run one by one. Each notebook writes its own set of output files, and the output files of one notebook become the input files of the next notebook. But for anything even a little bit complicated, this approach breaks down. We have to interrupt the linear sequence of notebooks in order to add steps. Intermediate files are cumbersome to micromanage, the code files get messy, and if you want to run tasks in parallel, it gets even messier. It's far too easy to lose track of what you're doing, and the end product is not computationally reproducible. Pipeline tools solve these problems. A pipeline tool orchestrates the overall flow of tasks from beginning to end, running the correct steps in the correct order, and if the compute resources allow on your platform, conditionally independent tasks in parallel. And for most pipeline tools, output data files are managed behind the scenes, so for each task you can trust that all the requisite data files are available and in the right place. Some pipeline tools even manage historical output, meaning they save output data from iterations of the pipeline that you ran in the past, and that's actually where I'm going for this talk. First, the targets R package. Most pipeline tools try to be language agnostic, and targets stands out because it works within the language. You can write complex pipelines that scale, but what you're doing as a user still looks like just using R. Everything from the abstraction of files as R objects to the way you can dynamically branch over dplyr groups or rows of a data frame to integration with R Markdown. And how it works is you write ordinary functions to prepare data and run models. You declare tasks that call those functions. Those will be your targets, your steps of the computation. And then the targets package actually runs the project. And it automatically knows which tasks depend on which other tasks, how to run those tasks in parallel, and how to store the data. On to how to get started. To use targets, you create what's called a targets file or a target script, which is just an R script that brings together the different components of the project. The use targets function actually writes a template targets file for you, and all you do is follow the comments and fill in the blanks. When you're done, it'll look something like this. So first at the top, you declare packages, options, settings, and functions, just as if you were setting up an ordinary R session for an ordinary project. And then at the end of the file at the bottom, you declare a list of tasks or targets that run when the project runs. Each target has a command, which is a bit of R code that calls your functions, and a name, which is a symbol. When the target runs, the command also runs, and the return value is assigned to the name. At that point, the target looks and feels like an ordinary variable in R, but it's stored as a file in a safe place. Remember that nice graph from a couple slides ago? Targets automatically generates it with the tar_viz_network function. The graph describes the progression of the pipeline in a natural flow from left to right, and it shows which targets are up to date and which targets are out of sync with the code and data they depend on. In this graph, one model is up to date and the others need a refresh, as well as the downstream output. When you run the pipeline, The correct targets run in the correct order, in this case distributed over 2 parallel processes, maybe 2 jobs on a cluster, and the output data is sent to storage. The deep neural net model is automatically skipped because it's already up to date, and the other 2 models automatically run to get back in sync with their upstream dependencies. And if those models change, the downstream targets will run as well. This selective skipping and running is an enormous time saver, and it creates a peace of mind that you don't know you need until you feel it for the first time for a complicated project. And that's what Targets cares about. But different pipeline tools care about different things. Targets is a lot like GNU Make, a classic pipeline tool from decades ago, which only cares about the current state of the project and skips tasks that are already up to date. But Apache Airflow is another key player in this ecosystem, and its priorities are totally different. Airflow cares about data pipelines that run on a regular schedule, that need to run end-to-end from scratch every time, and need a complete historical record of past results. Those features are valuable too, but they weren't always possible in Targets by itself. And that has to do with how Targets stores its data. If you've used Targets before, you probably know about the tar read function, which takes the file of a target that already ran and brings it back into the R session. The Targets data store is a folder that looks like this, and behind the scenes, tar read is going into this folder and looking in the objects folder for the actual data. The data could be a serialized R data file or RDS file, or it could be another format like Feather or Parquet. To learn what format it has and how to read it, tar read looks at the metadata file in the meta folder and then goes and reads the object. You can read the metadata file using the tar meta function. It has the name of each target along with the current hashes of the code, data, and dependencies for the most recent run of the pipeline, as well as other information like the random number generator seed used to run the command. There's also the path column, which tells us where each target is stored, and if it's a cloud target, the cloud bucket that it's in, and the version ID if the bucket is versioned. More on that soon. I chose this design for the Targets data store because it's simple, compact, resilient, efficient, and easy to understand. The downside is that it only tracks the current state of the pipeline. In the default scenario, there's no record of the past runs you did. However, the simplicity and the compactness open up a lot of possibilities for third-party tools to come help. Third-party tools that specialize in version control and can afford to help with the historical record keeping of the output data. The easiest plug-and-play solution for historical data management and Targets is integration with cloud storage. You can use Amazon Web Services Simple Storage Service, or S3, or Google Cloud Storage. Either way, you create a bucket and you turn versioning on. You declare the bucket in the options and you set the repository argument to AWS or GCP. Then, in the version control repository for the source code of your project, assuming you're using something like Git, track the lightweight metadata file in the Targets data store. That's the meta file in the meta folder in that store. The metadata tracks the version ID of each target in the bucket. Now you're prepared, you're set up. Continue to develop the analysis, run the project, and commit the code and the metadata with the target version IDs. And if you ever need to roll back the project, just check out an old commit of the code. The metadata rolls back with it, which rolls back the version IDs of the targets. In other words, when the code goes back in time, the data goes back with it, and your pipeline stays synchronized. Not only are the old results available, the targets are up to date as well. So you don't need to waste hours rerunning historical results just to get a reproducible synchronized pipeline again. Here's how it works in a target script. Most people usually set the bucket in tar option set and the repository in tar target, but you could do both in one or both in the other, or in fact use different buckets for different targets, even sending some targets to Amazon and other targets to Google. On the other hand, cloud services are a steep learning curve and they could get expensive depending on the use case. For a completely local solution that doesn't use those cloud services, the Git targets package is another way to track output data over time. Here's how it works. First, run the pipeline and commit the code to version control with something like Git. That much is standard procedure for a targets project. After that, we go to Git targets and we use tar git init to create a repository for the targets data store. The data repository is separate from the code repository because there's usually a lot of data and we still want to be able to push the code to GitHub or GitLab, so it needs to be light. Now comes the main part of Git targets. When you reach a strategic milestone in your project, when your targets are up to date and the pipeline and your code is up to date in Git, you can use tar git snapshot to commit data files. The data commit makes a special reference to the code commit that was active at the time. So if you ever roll back the code to this commit, then tar git checkout rolls back the data to match that code. At that point, not only do you have historical targets in your workspace, those old targets are up to date with the old code that you just checked out. And now to walk through a demo. Let's say we've already run the pipeline and committed the code to version control. The next step is to create the data repository. When that happens, it tells you what it's doing, including some Git LFS configuration. As we go through the work of developing the project and we reach a strategic, important milestone, we make a data snapshot. When we call tar git snapshot, Git targets creates the special code equals branch that has the SHA key of the currently active code commit. Those code equals branches in the data are how code commits map to data commits. Each data branch corresponds to a code commit where you made a snapshot, and only the latest data commit in each code commit really counts and is important. For a historical record of the data snapshots you made, use the tar git log function to see which code commits in the current code branch have corresponding data snapshots. And then to roll back the whole project, you can check out an old code commit and then use tar git checkout to retrieve the old data that matches that old commit. If you committed and snapshotted an up-to-date pipeline before, the checked out pipeline will be up to date as well. Finally, I'd like to thank Mark Edmondson for implementing the internals of the Google Cloud Storage backend and targets, and I'd also like to thank the rOpenSci reviewers of targets and Git targets, as well as rOpenSci as a whole.
Thank you so much for your talk, Will. Anybody has questions? We have a little bit of time, so if not, we can move on and we can ask in the end. So our next presenter is Ilias Moutsopoulos with bulk analysis in R. Thank you so much. Floor is yours.
Right. Hi, everyone. So my name is Ilias. I'm a bioinformatician at the University of Cambridge. I'm doing my PhD at the Stem Cell Institute. So it's a bit of a shift from what Will was talking earlier. And I'm going to talk to you about Bulk Analyzer, which is a package that we developed over the past year or so. So if you want, you can scan the QR code and go into a live version of the app, and that'll be relevant in a few minutes, but it'll reappear a bit later. So Bulk Analyzer is a pipeline for analyzing and sharing bulk sequencing data. So I know I'm not talking to biologists or bioinformaticians, so I'm going to start at the beginning. So what is sequencing data? So when you have a hypothesis, you want to test it. As a first step in the biological side, you can perform RNA sequencing, and you can essentially get copy of the RNA that's within the cells. You can go through a protocol to sequence it. DNA and RNA consist of bases, ATCG or AUCG for RNA, and you do that experiment and you end up with these really big files called FASTQ files that have each line represents a read, so about a length of 100 base pairs, and you have these huge files of like 5-10 gigabytes each with just reads. So the first step to process that data is kind of the standard bioinformatics like processing, and you can do some quality checks. You can align it to the reference genome if it's human data or mouse data or whatever it happens to be, and then you quantify the genes, and every gene has an expression, which is the number of reads that aligned along the sequence of that gene. So you end up with an expression matrix, which is M by N matrix, where you have genes on the rows and samples in the columns. So usually in an experiment, you might have from 2 to like hundreds of samples. But the idea is some of the samples are in one condition, some of the samples are in another condition, and you want to compare the two conditions across all the genes. So downstream processing can be many things. You can start with quality checks and do some more statistical analysis and downstream interpretation of the results. And there's various tools and pipelines and various ways to perform that. And exactly what you use may depend on the experiment or the hypothesis that from the biology side. Now, the problem with that is currently downstream analysis it branches into two. So it can be either very rigid and done automatically. There are many services that do this sort of thing. Some are paid for and are better, some are not paid for and are slightly worse. But the general principle is there are some standard steps. You plug in your data, you get an answer, you get some plots, but you cannot really influence any of the steps or any of the parameters, or if you can, you have to specify them at the start, so you might have to rerun the whole thing again if you want something slightly different. And generally that's good for a like first look at someone's data, but it doesn't go much further. It doesn't go into detail and towards like proving a hypothesis or a publication. Now the other side is collaborating with a bioinformatician like myself. So the biologist comes and gives you the data, you do some first analysis, you give it back, they have a look at it, they give it back to you, they say, oh, can you change that, can you add this? And this whole process, as you can imagine, takes a long time and can cause major delays in the projects, especially because the biologist might have an idea but can't immediately test it, and they can't directly see the difference that their idea is causing in their data. So to kind of be somewhere in between those two extremes, we've developed Bulk Analyzer. So Bulk Analyzer is an R package that is based on Shiny. So Shiny is a way to create web interfaces through R without going into the HTML, JavaScript, and all that. So the idea is we have some first steps to process the expression matrix and the metadata. So metadata is just referring to information about the different samples. So you might have time or some other condition, so control versus treatment. So you have that information tabulated. So you do some pre-processing, and then you create a Shiny app with bits of analysis that you're interested in. So the Shiny app may have like quality checks, some downstream analysis, some interpretation, some network predictions, but it's all interactive. So you can create that app with just a couple of lines of code. There is the pre-processing function, and then there's the generating function. And there's many parameters you can specify to like change what the app looks like, and specify which organism you're looking at, which tabs you're interested in, and the general structure of the code. Now, because the app is generated. The app is generated as a standalone document, so it just generates an R script and some RMD object, not RMD, some R objects. So you can then host that app. You can give it to someone else, and they can just run it and look at it independently. So if you want to have a look at the live app, you can have the QR code from earlier, but let's start by the pre-processing. So the first step in pre-processing is noise removal, and that's a whole different project that happened in the last 2 years. But essentially, we've developed a different R package called NoisyR, which is also on CRAN and can be used in the same way. But to remove noise from the data, to make sure we're not biasing downstream interpretation by low expression distributions that do not make are not reproducible, or just technical variation that may not appear if you did the experiment again. And the second step of pre-processing is normalization, which is a standard step in RNA sequencing. We have many options, basically all the options that are available in the literature for normalization of RNA-seq data. So once you run the pre-processing, you can create your app, and an app typically looks like this. So you have a landing page with some links on material to understand how it works, a link to our preprint, and each of the individual tabs are tabs that have been selected by the app generating function, so you can influence what tabs you see. But going through the tabs, you can perform a standard RNA-seq analysis. But you can do that in the Shiny app, which means you can change any parameters interactively. You can modify your plots, you can modify your output tables, you can download those plots and output tables, and these are good quality plots that you can then directly input into a manuscript, for example. So the idea is to avoid having too much interaction with a bioinformatician to produce kind of the standard results that you would need. But going a bit beyond that to generate hypotheses, modify your results, and it generally speeds up the process of analysis. So again, if you want to see a live version of this app, you can scan the QR code. So the app contains what we've experienced as the main steps of analysis that bulk RNA-seq are usually used in bulk RNA-seq. So we start with some quality checks. We have some heat maps to understand like the structure of your experiment, the principal component analysis, again, to see how your data groups together, if what you expected. So this particular data set is 3 time points with 2 samples each, and you see that the time points separate nicely. And then we have some tabs for downstream analysis, so differential expression, which is a statistical test between 2 conditions, so for example, control and test, to see which genes are different between the 2 conditions. And you have a large amount of freedom to play with the options in these plots, change various thresholds, you can click on the different genes and see what happens, which ones they are, you can label them, and you can download these plots. You have the gene heat maps, you have genes on the rows, samples on the columns, and you see the differentially expressed genes are expressed differentially between the 2 conditions. You can get a time series, expression patterns of different genes plotted as line plots. And you can do some more stuff. You can do enrichment analysis. So take your list of genes and plug it into a database, and which tells you which biological terms are enriched from that gene set. So if your genes are similar to each other or are completely different, and you just have no interesting processes there. You can have cross plots, which are essentially comparing 2 comparisons against each other. So things around the diagonal means the comparisons are similar. But we've gone a bit beyond that, and we've added a gene regulatory network component. So gene regulatory networks are the network of interactions between different genes and transcription factors in a cell these can be visualized as a network. So an edge between two genes mean that these genes interact. So one is the regulator and the other one is the target, for example. So we've added this module into the package to generate these gene regulatory networks with a center around one gene to try and understand some of the interactions that happen and try to guide the biological analysis. And you can have multiple ones of those, you can compare them. And the other thing we added was you can have extra information. So except for the information on the genes, you can have cis or trans interactions, so either proximity-based information or correlation-based information, and you can decorate the networks using that information to get more out of your visualization. So for this, for example, is positions in the genome where you have open chromatin, but that's a detail that's not important in this context. It's that you can generate those networks dynamically. You can choose the genes, a number of edges, and you can interactively play with your data as the biologist, so you can generate more robust hypotheses. And the app can also supports multiple data sets and integration of different data sets with each other. So that's Bulk Analyzer. Thank you very much. And I'd like to thank my co-authors and the rest of the members of my group and my collaborators from the Stem Cell Institute. But I'm happy to answer any questions.
We have a question on the Q&A, and the question is, how do you distribute the Shiny app results for a collaborator to explore? Or do they need to install R and run Shiny to see the results, or is there a web link in a server?
That's a good question. So that depends on the personal infrastructure that you have. So you can do both. Personally, we have a website that we use to host the Shiny app, so they don't need to install R and Shiny and have all the dependencies ready. You can use a container to have everything pre-installed so that there's no versioning issues, or you can just have someone who has some experience with R just download Shiny and run the app through that. It depends on what you have available and how familiar your collaborators are with R.
Okay. Do we have more questions? Wait a little bit. Okay, it seems that no more questions. And think about the question later. You can also put it on the Q&A and answer them after all the talks are done. So our next speaker is Oliver Heide. That's going to talk about providing large trait data sets for research using Apache Arrow. So the floor is yours. Thank you so much.
So, hello everyone. Good morning or good afternoon. Welcome to talk about providing large data sets for research using Apache Arrow and Parquet. My name is Oliver Reiter, and this is joint work with my colleague David Zenz. We are both working at the WIIW, which is an economic research institute. Since one of the research focus of our institute is international trade, international economics, international trade data is used in many projects. There are two very commonly used data sets, for example, the UN Comtrade or the EU COMEX, which is a monthly database, but also tariff data and so on. They all have in common that they are very large in size and usually larger than memory. For example, there are more than 200 reporting countries. One says exporting country, whereas importing countries, exports, imports, re-imports, re-exports, and so on. So the trade data is really very, very large. And we have several use cases in our institutes. Either we're interested in very specific queries, so queries for specific countries, flows, years, or products, or we aggregate the trade data, for example, calculate bilateral trade flows, so we sum over all products. But for both use cases, we need to have the detailed trade data available, locally available. In our previous setup, we downloaded, processed, and saved the files using several data scripts, which were run consecutively. And right now, we are using or moving our pipeline to use more of Arrow and Apache Parquet to economize on storage and make also queries more efficient. So what is Apache Arrow and Parquet? Apache Arrow is a platform for data analytics, and is a cross-language development platform for handling flat and hierarchical in-memory data, and is organized for efficient analytical operations on modern hardware. It also provides a lot of language bindings, for example, C, C++, C#, Python, and of course also R. There was a major release recently, the version 8, which comes with a lot of new features, and I encourage you to check out the homepage, which provides detailed changelog. Apache Parquet is a column-oriented data storage format and provides efficient data compression and encoding schemes to handle complex and large data in bulk. In this presentation, I'll use the Apache Arrow and Parquet names somewhat interchangeably, which is not correct, but for the presentation, I think it's fine. So just go quickly over our use case for large databases. And so recently we had, for example, a local copy of the UN Comtrade database available, and we saved the trade data into a huge number of separate files. So we created a directory structure that was separated by database, by product classification, by flow, but also by report and by year, and then had a file saved in there. So when you needed to access a certain subset, you had to write your own loop, sort of going over all the files, loading them, extracting the data you needed, compacting them, and save it somewhere else for further transformation. That resulted in long reading and actually also long writing times, and made updating the database, which is done usually twice a year, very, very cumbersome. So there we had actually several scripts, and each of those scripts was essentially a loop that read a file from somewhere, applied some sort of some transformation, for example, moving the country classification from one classification to another, and then save the file somewhere else again. And as you can imagine, this was very a long-running process. So right now, we're moving to Arrow and Parquet, and using Parquet for example, for storing the data on disk, which is efficient in two terms, in terms of size and also in terms of accessibility, which you'll see later on. And starting with Arrow version 6, computations can also be delegated to Arrow. There you'll write a transformation pipeline using dplyr verbs, and Arrow actually executes it for you. So there's no need for you to load the data into R, actually to load them into memory. and do the transformation into R itself. It also allows you to write a data set in one step, and the Arrow actually takes care of the partitioning you want to have. So that allows you to write very high-level code for the whole database, more or less, rather than low-level code for individual files that we used to do before. So just a quick example, the EU COMEX is also a trade data distributed by Eurostat, and is actually available for every month. So we now built a pipeline that runs on every 20th of the month, checks the website if there are new monthly files available, and if there are new files that we don't have locally, we actually just do a loop, download and read the files, transform, and write them out as Parquet files. So you see a very stylized version of the script here. It's not much longer than that, actually, and makes the whole process very easy. So with that, we can already mirror the EU COMEX as it is available online, locally to our disk, to our server. And actually, the EU COMEX is available monthly, but we also want to access, for example, yearly data, and for that we can actually use one of the features that were recently introduced to Arrow, in that we delegate the computation of the aggregation of the monthly data to yearly data to Arrow, and there's no need to do the aggregation in R itself. And you can see here that the stylized pipeline that we're using, and actually updating 1 year of this runs in actually in 18 seconds, which is very, very fast if you think about it, the amount of data that you actually have to read in. Then again, writing out this new computed data set with another command makes the whole process very easy. So the whole process is actually just two scripts that are run to each other, and allows us to keep a monthly and a yearly version of the EU COMEX locally available. So I've talked a lot about Parquet, but what are the numbers? For example, for the EU Comtrade, we actually did a comparison how the storage sizes differ. If we would write out the whole database for several years, for all years, for all product classifications, for all flows on disk, the CSV files would amount to a little bit less than 1 terabyte. The data files would actually be more than 1 terabyte. However, if you do that all in Parquet, it only sums up to 55 gigabytes. So that's size reductions of more than 90% if you move to using Parquet instead of these other versions. And I also hinted at it, that we're not only interested in storing it efficiently, but also accessing it efficiently and do the querying efficiently. So that's why we did a sort of benchmark setup, a simple testing of which file formats and accesses perform the fastest. So we did this on a Debian server. We used a relatively recent R version, but the tests we did were a year ago, and there was the Apache Arrow version 3, the current one. Right now it's 8, so we only expect Parquet and Apache to do better now. We use a subset of the EU CompTrade, and the first table gives you here an overview of the file sizes that are generated by a few different formats. So here we not only compare comma-separated files or starter files, but also the RDS file formats that are included in base R, and also the FST formats from the FST package that uses the also a very fast compression algorithm. And you can see Parquet files are actually also the smallest on disk. And the table below here gives the smallest on disk are actually the RDS files with the highest compression ratio, and second is Parquet, and the table below gives the sizes, the relative sizes. Then we tested 5 different queries that were from simple to complex. The query 1 is very simple, where we just extract 1 year of one exporting country and one importing country, and then make the queries more complex up to query 5, where we actually access several years, several exporters, and several importers. They are designed to imitate the practical use cases in our institute, and we also extract different subsets. So not only we also do a subset by flows, a subset by reporters, by reporting countries, or partner countries, and so on. And the table below gives the timings of the different formats. And you can actually see what the biggest take-home message is that Parquet performs very steadily and is very immune to the increasing complexity of the queries. In queries 1 and 2, it's actually not the fastest access method. So there you could do better with choosing other formats, but that's only the case because those simple queries actually coincide with reading one single file, and that's actually a little bit faster than Parquet, which also has to do a scan over the file system. But as we increase the complexity of the queries, all formats and take longer and longer, and only Parquet and Arrow and Parquet is more immune to those increasing complexity of the queries. So coming to an end, what are the advantages of using Arrow and Parquet? It drastically simplifies the setup of keeping your database up to date. It actually hides from you the distributed file system that it uses to store those files, and it enables the users to write very high level and very understandable code because you can do you can use the dplyr verbs that act on the whole database rather than on individual files. For Apache Parquet, we see massive reduction in file sizes, of more than 90%, and it allows actually to store a lot, to store big data sets on modern PC hardware. However, one has to choose the partition of the data set, and that is a bit of a question how to do it best, because you can choose that partition or that partition, but it very much depends on what queries you usually want to execute on that data. And so to minimize the number of files, the Arrow has to touch to extract the needed data. And for Apache Arrow, it actually outperforms CSV in the bigger and more complex queries. And the execution time of Arrow and Parquet actually stays constant, while the execution times of the other formats actually drastically increase, and it offers the best trade-off between the timings and the file size. With that, I'm at the end of my talk. Thank you very much for listening, and looking forward to your questions.
Thank you so much for your question. I'll be taking questions. Does anybody have any questions? I see one question. Waiting a little bit.
Or maybe afterwards.
Yes, maybe afterwards. So let's move on with the next presenter. Phong Quang with Dayquery. So welcome. The floor is yours.
Hi, just let me share my screen. It's an interesting effect. So, hello everybody, my name is Phong Quang. I'm a medical statistician and a part-time DPhil student at the University of Oxford. I'm going to talk to you about my new R package called Dayquery, and what it does is it creates data quality reports for temporal data sets. I'm going to talk about it in the context of electronic health records and their use in research, because that's the field that I work in, but they can it could equally be used in any field that uses temporal data sets. So what currently happens as a researcher when you receive a new data set, you need to check that it contains what you are expecting it to contain. This is done in the initial data analysis stage. You may check for missing values, remove any duplicate records. I may run some summary statistics, and I may check for look for any outliers if I'm sort of going to be building a model or something. Essentially, I need to check that my data is actually sufficient to use for the analyses that I plan to use it for. Problems with this is that it's all completely ad hoc and undocumented. It's said that 80% of researchers' time is spent on this sort of data cleaning stage, but it's never reported in published papers. So you just have to assume that the researchers are doing it, and sort of fingers crossed they haven't missed anything. Another issue is we're getting more and more data. The more data you have, the more checks that you need to run, and sort of finding the time to do this sort of fairly unglamorous part of the analysis process. And another sort of issue specifically relevant to routine data sets is that the actual sort of process that generated the data, you're so far removed from it that you have no control over it. You don't actually know what's happened. And if you've got a long data set, that's been collected over a long period of time, things will have changed during that time that you're not aware of. And so it's sort of incumbent on the researchers themselves to check for these sorts of things. A few examples of what can go wrong. So this is real data from a large UK hospital. In the first graph, it shows the number of inpatient admissions per month over a period of 25 years. And you can see that in around 2008, this number shoots up quite remarkably, and then it sort of drops really suddenly in just before 2012. Essentially, the way that the admissions were recorded was different during that time. If you're going to be using this data, you need to know if there are certain periods of your data where you need to make adjustments to ensure that it's consistent for the analysis you're conducting. The second example is from a laboratory where the test, so this is the minimum value for a creatinine test, which is a blood test for a marker of infection. And you can see that there's sort of 3 distinct epochs of data, or of the values that have come off the machines. And essentially, so in before 98, there's sort of a bimodal distribution, which was caused by the fact that the values were being recorded under 2 different units. Then there's sort of a period of sort of good stability, and then in 2009 there's this sort of sudden shift downwards, and it's clearly not the patients that are changing. It's clearly some sort of system issue here. And what this actually was that the test used to generate these results was changed on that date. So if you're a clinician in hospital, you would have been told that we're using a new test. These are the new values that we're expecting. And so you adjust your management of the patient accordingly. But if you're a researcher receiving this data set 10 years later, you're not going to be told that. So you need to make sure you're checking for these sorts of things yourself. So what DAQRI does is it takes basically a generic data frame. It specifically is designed for temporal data sets. So the only actual sort of condition on your data set is that there is a date field which represents the date that that particular row of data corresponds to. Whereas the rest of your data fields, which typically would be other bits of information relevant to the event that's being described in that row of data, can be, you can have any number of different types of data in there. And so when you're starting with this, how do we sort of look for potential data quality issues? What the DAQRI package does is it generates for you a range of temporal visualizations of the data. So here you can see very clearly that across the 1 year period that the data covers, in July, the number of records increases quite substantially. And you can also see quite clearly that in the month of April, there were no records at all. And so this gives you a very quick and easy sort of view of potential issues with your data. So I'm going to give you a quick demo, live demo. So the package is currently under review at ROpenSci. If that all goes well, it should be going on CRAN in the not too distant future. But in the meantime, you can install it from GitHub. I've already done that. So if you attach the package, there's help documentation for each of the functions. I want to point you specifically to this vignette, which is the walkthrough, which describes in detail how to use the package. So we'll go through that immediately, but that's the best way to understand how it works. So I'm going to go through an example data set that's actually supplied with the package when you install it. It's basically a CSV file. When we load up this data, so we need to load it up into a data frame. You could use whatever sort of file reader that you typically use, but the package offers a helper function, which basically reads in your data all as character data fields. And the reason for this is that DAQRI will check for any non-conformant fields. If you're expecting a numeric field and there's values in there that are not numeric, it will collate all those warnings for you and present them in the report. So I'm just going to run that quickly. So this particular data set has 9,000 rows in it and 8 variables. We can look at what's in it. So this is basically the data set that I've shown in the slide. The next thing we need to do is to specify what sort of data is in each of the fields. So as I said before, the most important field is the time point field. So which field represents the actual sort of what you want to be your x-axis on your graphs. So here, this is the prescription date for this example data set of antibiotic prescriptions. Now, with each of the other columns, it could be a date field, which may or may not include a time element. It may be a numeric field, it may be a categorical, or it's actually a factor field. And in this case, we can additionally sort of ask it to generate some additional graphs per category in that field. So the location has site 1 that's visible there, but we'll get extra information for that. So I'm just going to run this. So that's created the specification, and then to create the report, the easiest way is to use the create report function. Takes the data frame, specification, and there's a few extra parameters available. One is any sort of string values that you want to be treated as missing values. Here we've got a number of nulls in there, and so we want to make sure that it identifies any nulls as a missing value. The other important thing is specifying the level of granularity that you want to be applied to the data. So by default, it's aggregated by day, but you could also specify it to be done by week, month, quarter, or year. I'm going to save it in the current folder. I'm going to print the progress to console so we can see what's going on, and then also have the option of recording the progress to a log file if you want to be able to refer to it later. I'm just going to run this right now, and you can see it running. So it's aggregating the various data fields depending on what you've what type of field you've told it is. Now it's knitting the markdown report, and this will be individual to your data set. And it shouldn't take very long. It's almost there. And then it's been saved. So here it is. It's popped up. And if I show you the report, so there are a few tabs. The first one just gives you a bit of an overview of the data that you've imported. You can also get the results of any validation warnings. So I've said that the data field should be a number, but there's certain values in there that aren't numbers. So it's sort of creating all of these and letting you know. You've also got a few sort of overview plots. So this is one I showed you earlier, which is the number of records and the number of values in each particular data field in your data set. You've also got overviews for the number of missing values, any non-conformant values, duplicate records. If you want to look in more detail at individual data fields, you've also got one tab per data field in your data set. And all data fields will generate a graph for the number of values as well as the number of missing values and the percentage of missing values. But depending on how you specified the type of field that each field is, you get additional tabs for different things. So here, this was a unique identify field. So we're looking at sort of the length of the string to see if that changes over time at all. Another example, so the location field, which was a categorical field, we might want to look at the number of distinct values in that field. And we see that it changed from 1 to 2 in July, and we told it to sort of break down those values per site, sorry, per categorical value. And so there were two values, there was site 1 and site 2, and you can see how these values change over time within that particular field. So site 1 has been there sort of the whole time, and but then site 2, you can see this value only started coming in July. So I'm just going to switch back to the presentation. So it's this one. So why should people use it? We want to encourage good research practice. Essentially, we're getting more and more data. It's important that we check this data is what we expect it to be in order to create better science, basically. It increases transparency. So the reports is just a sort of self-contained HTML page, and so you can easily share this. You can attach it to an email. You can upload it as supplementary information to a published paper. And it's hopefully, I've shown it's easy to use. I think the main benefit is that it works on a generic data frame. Your data doesn't have to be in a certain format. I've sort of demonstrated it in terms of a raw data set, but it could equally be run sort of after data cleaning or sort of after reshaping. And you can also share it with collaborators, people who don't necessarily know how to use R, and it's very easy to see what issues there might be. So lastly, I just included a few links to the repository, and there's a package website where you can browse through that example report that I quickly showed you, and the slides and the demo script are also available on GitHub. So thank you. That's the end of my presentation.
Thank you so much. We already have a question for you, which was about the links. And if you could drop them answering that. And then we have another question is, does it support ways to identify outliers, breaks in the time series other than graphically? So I guess if there's a reference method for that.
So the original sort of goal when I started building this was to find sort of automatic methods to identify these sorts of what I call change points. Unfortunately, the methods that are currently available did not work very well at all on sort of real world data and on the sort of the range of different types of data sets that you can get. So we found that actually the current state of the art isn't good enough to sort of do that. But the human eye is really good at doing that, and so just sort of making it easy in any sort of way, just even though you still have to click through the different tabs, it's much quicker to generate and click through the tabs than having to generate all these graphs yourself.
Okay, and then is that is more in the document that you can keep track of things that change, such as units, and perhaps a collection practices change for the entire data set?
So there's nowhere in the package where you can document that. That's another question. That's a good question, especially when we're reusing data sets. You might find something and then you want to tell somebody else that this is an issue. I don't currently have a mechanism for that, but that can definitely be something to be thought about in the future.
Maybe implement something similar in the future? Is that something?
It's a possibility.
Maybe. Okay.
Maybe.
Okay. Cool. And I'd like to know if the reports of these documents can be generated in LaTeX format. Or
Not at the
PDF?
moment.
Okay.
So I've concentrated all my time so far just getting it out on HTML. If there's enough demand to get it into LaTeX, then that can be considered. But as I say, it's only just being put out into production now. Yeah.
Okay. Very cool. Then we have a question for Oliver Heiter. We have two. The first one is that error mostly leads for queries in the connection, or you can also do that with Spark and the algorithms.
So to my knowledge, there's no, or at least not yet, a link to the mlib libraries. So there are a few competition engines being developed, but right now they concentrate on more or less high level or sort of simple functions that can be delegated to Arrow. But I don't know. I don't think that there's a connection to Spark yet.
Then we have another question, which is, so this person has a vector column that's very large, and she wants to partition it over. Very high cardinality, and you need to filter on this specific vector very often. There are a few less than 400 important levels, and all the remaining ones could be saved in a specific partition if possible. Is this type of functionality available in Arrow? Or is it being, if not available, is it being developed?
Mm-hmm. So I'm not a developer for Arrow, so I don't know. But if the cardinality is very high, and that's usually a problem, or so you should try not to create a file structure that is too diverse, too wide also. too hierarchically deep and nested. So as you suggested, if you have a few very high important factors that you often subset with, and the other 29,000 you don't care about that much, then why not sort of create an aggregated version of the factor and separate by that. So you had 501 factors, and that would be possible, yes. So you should find something that allows you to separate the factors on a more hierarchically higher level.
Okay, cool. Was a question for chat about the Shiny app, about how long did it take you to develop it?
Yeah, so it kind of started as a on-the-job project, essentially trying to write some functions to automate some of the tasks that we were normally doing. And so if you count that in, it probably took about a year in total. For the first maybe 3 or so months, I did some stuff by myself, and then I got Ellie, my collaborator, in as well to actually put it into the app. And that took a few months to make sure everything is linked up correctly and the Shiny modules are self-sufficient and everything works well with each other, and it's extendable to adding new things as well.
And I that too. Is it easy to maintain now that you have set that up?
It's very easy to maintain. The main issue with maintaining is dependency on other packages.
Oh, I
But
see.
the code base itself is very nicely laid out, because it's each tab is within its own module, and input and output is controlled very nicely. And it's very easy to add new modules as well, if you want to add a custom tab.
Okay. There are a lot of questions for Will, but Will has been much faster than me and has been answering them on the chat. So the last one was if the pipeline contains both R scripts and external softwares, if Target could contain another one. And so if you want to expand a little bit on the questions you've been having, then it's very nice. But then you can have some time to explain in more detail.
Sure. So each target encapsulates at the highest level of a pipeline what a piece of work that goes on and returns a value, and anything can happen within that target. Usually, most successful targets return an R object in R, and have few side effects. What happens to get there to that return value is entirely up to you, and so you can invoke functions in R that call external tools. So there are a variety of ways to do that in R code. And then this becomes, because every target has arbitrary R code, the issue of calling external software is actually one that's more of an R issue than a targets issue. And so there's the system2 function in base R to call system commands. There's the process X package by Gabor Sardi from RStudio to have a more portable interface to call external software. Part of that external software can include Docker. And there's this excellent blog post by Joel Nida, and he's a longtime target, and he's done a lot of bioinformatics workflows that use Docker. I'll link his post to the chat. He calls within a target, he calls a package called Babel Whale, which connects to and calls Docker instances that can run a command and produce an output. Very possible. Anything you can do in R, you can call from a target. So I don't personally use Docker a whole lot, but that specific pattern is promising.
Oh, it's very cool. Thank you so much. Then I have another one from Ilias. Can you share the URL version and then are you planning to expand this package to single cell analysis? So I guess the Shiny app to do the same type of analysis for single cell.
Sorry, what was the first?
If you could share the link to the QR code,
Oh yeah, absolutely.
and then if you're planning to expand it to single cell analysis.
So cell analysis, there already exists a kind of similar concept that's called Shiny Cell, and it's
Okay.
that's actually what gave us the idea of developing something similar for bulk. So yeah, so
Okay.
it's a very nice package.
So no need to further developing that. It's very cool.
Yes. Yeah, so we're basically going to work on this and add modules expanded based on feedback from users.
So do we have any more questions?
This is the link.
Still have a little. These were everyone. Thank you for joining us here today. It was a very good talk. A lot of promising work. We have to keep updated with everybody's work in the future. And yeah, for now, we would love to see how the project over time. When you get to see when a project's starting or like the first steps, and then you look back over it. As always, no questions on the chat. So again, thank you everybody for being and joining us here today. Thank you for the speakers for the talk. And again, thank you for sponsoring the event. So again, thank you so much. Bye-bye.
Bye.
Thank you.
Bye bye. Thanks for sharing.